And now we conclude dramatic discoveries. How is new DNA made? First, you learned about Meselson and Stahl's hypothesis that explains how new DNA is made. You remember Dr. Meselson and Dr. Stahl? Their hypothesis is called the semi-conservative hypothesis. It says that new DNA is made by combining one old strand and one new strand to make a single new molecule of DNA. Next, you came up with an alternative hypothesis of your own. Meselson and Stahl also had an alternative hypothesis. It is called the conservative hypothesis. It says new DNA is made by creating two new strands that come together to make a single new molecule of DNA. The original molecule of DNA is left unaltered. So what happened next? Which hypothesis was correct? To find out, Meselson and Stahl carried out a classic experiment. They put special labels on the new and old DNA strands. We will call it a heavy label on the old strand and a light label on the new strands. If the original semi-conservative hypothesis was correct, they expected to see only one type of DNA, an old heavy strand mixed with a new light strand. This makes a medium-weight DNA molecule. But if the alternative conservative hypothesis was true, they expected to see two types of DNA, old molecules with two heavy strands and new molecules with two light strands. The first time Meselson and Stahl tried their experiment, they had to throw out the results because Meselson wrote the wrong labels on their tubes. No joke, this actually happened. Undaunted, they repeated their experiment a second and a third time. Both times, they found that the new DNA molecules were made up of one heavy and one light strand. This supported the semi-conservative model. Thanks to this hypothesis and experiment, scientists and doctors have made many innovations and further discoveries. Things like cancer drugs, antibiotics, and modern biotechnology are all possible because of the knowledge learned from this experiment. So remember, a strong scientific hypothesis is an essential part of the scientific process.